Professor Shin, for sharing your amazing work and contribution to the field of viral hepatitis. So if I may, we will now move on to my talk. So my name is Edmund Neo, located in Singapore, and I'm the sales representative for ProImmune in Asia. I've joined ProImmune for about 2 years after graduating from the National University of Singapore. So today I'll be sharing ProImmune's capabilities that we can offer for epitope identification and clinical immune monitoring projects. Here's an overview of the capabilities that ProImmune can offer, which I will be sharing on our MHC multimer technology, which we have seen quite a number of data that's been generated by Professor Shin and later on by Antonio Bertoletti as well. So, and then we have more than 20 years of experience providing to our customers for the multimer technology and has been cited in thousands of papers, including by our speakers today. Next, I will be also sharing about our REVEAL MHC peptide binding assay that we offer as a service to screen peptides against MHC alleles to assess binding characteristics. Using a combination of tools allow you to save time by rapidly identify and validate epitopes for immune monitoring programs, which enable to assess efficacy for vaccine and immunotherapies programs. Our MHC multimer technology enables a robust technique to evaluate function and frequency of immune responses, which allows you to draw correlation between treatment and clinical outcome. So before introducing our MHC multimer technology, I would like to first talk a little bit about the role of the MHC molecules. So most of you might be familiar with the MHC molecules, but it's still important to explain for us to better appreciate our MHC multimer technology. Here I've drawn a simplified cartoon where we have the MHC molecules classified into the MHC class 1 and class 2. The MHC class 1 consists of 3 alpha chains and 1 beta microglobulin, while the MHC class 2 consists of 2 alpha chains and 2 beta chains. Here we have the antigen presenting cells, which can undergo proteasomal cleavage pathway expressing MHC class 1 and endosomal cleavage pathway expressing MHC class 2, presenting the antigen on the antigen presenting cell surface. Then we have the T cells where the antigen-specific CD8 T cells, T cell receptor recognizing the peptide MHC class 1 complexes, also known as the cell-mediated immune responses. On the other hand, the antigen-specific CD4 T cells, T cell receptor recognizing the peptide MHC class 2 complexes, also known as the antibody-mediated immune responses. Now with that, the next question is, how can we detect the antigen-specific T cells population? You can do so by deploying our Pro5 MHC class 1 Pentamers to detect antigen-specific CD8 T cells. Let me explain the beautiful design of our Pentamers, which consists of 5 MHC peptide complexes that are pentamerized and assembled through a coil domain. Each pentamer also comprises up to 5 fluorochrome or biotin tags for bright and efficient labeling. Due to the planar configuration, all 5 MHC peptide complexes on the pentamers are available for binding to the complementary T cell receptor. Therefore, the pentamers binds to the antigen-specific CD8 T cells, and with the fluorochrome conjugated, you can then perform flow cytometry to look at the antigen-specific CD8 T cell population in your samples. So for our pentamers, has been widely used by many experts in the world and has been cited in thousands of publications. We are the world-leading multimer manufacturer and provider evidently shown in our publications track record. If you have any questions about using our products, we have a team of experts to assist you very quickly to address those questions for you. Our Pentamers gives you additional flexible labeling and storage options with the choice of labeled or unlabeled format depending on your project requirement. We have an extensive catalog list which consists of 49 viral hepatitis specificities and 20 SARS-CoV-2 specificities that are ready to be shipped out in 1 to 2 days or 2 weeks. If the specificity that you want is not found on your catalog list, we can provide them as a custom pentamers where we have the mouse, human, and non-human primate alleles available and ready to be shipped out in about 4 to 6 weeks, depending on the binding affinity. So we offer the pentamers in the pre-labeled option that comes in the RPE, APC, or biotin, which has a long-term storage of 6 months at 4 degrees Celsius. We can also provide the pentamers in the unlabeled format, which the Pro5 fluorotext will be provided separately for you to perform the conjugation before the usage. The advantage of the unlabeled format is that it can have a longer term storage of 12 months as you can freeze the pentamers down at -80 degrees Celsius. Another advantage is that you do not have to decide in advance which label to use for the whole batch of pentamers for your experiment. They are all offered in 50, 150, or 500 test quantities, with larger volume ordered at once being more cost-effective depending on the scope of your project. To order our catalog pentamers, simply follow these 3 steps. Firstly, visit the web link below, which will bring you to our website showing the full range of MHC-restricted epitopes from various origins. Secondly, if you have any specific research area or epitopes that you are looking for, type in to the search bar pointed by the red arrow to filter. Lastly, contact us to place your order and your pentamers will be shipped out from the UK in 1 to 2 days or 2 weeks. However, if you cannot find the pentamers in our catalog list that you would like to have, we can provide them as a custom pentamers for you. Here is a list of MHC alleles that we can provide for the custom synthesis, and here are the 3 simple steps for you to customize your pentamers. Firstly, we will need you to provide us with your desired MHC peptide information, which our experienced team will assess using a combination of different tools to check if we can accept those sequences for a custom synthesis. If not, we will request supporting information from you. Secondly, once the MHC peptide has been accepted, you may place the order with us and we will start with the pentamer synthesis. Lastly, QC will be done and shipped out in about 4 to 6 weeks from the date of your order. If you have any questions about using our pentamers or you would like to get a better staining data, here are some common troubleshooting tips of using our pentamers, which usually helps our customers a lot. Firstly, remember to centrifuge the pentamers before usage, which this has been commonly missed out. Secondly, after doing a round of pentamer staining using our suggested parameters and you would like to optimize your staining data, you can find optimization protocol in our Pentamer Handbook, such as by changing the incubation temperature and time and by titrating the pentamers. We also have examples of showing a proper gating strategy for live lymphocytes and to perform a proper compensation. If you would like to receive a hard copy of this Pentamer Handbook, feel free to let me know and I am happy to make the arrangement to send you a few copies right to your doorstep. If not, you can download the PDF version in the link below. Now I'll be sharing a case study on the success for HCV vaccine trials at Oxford University by Ellie Barnes and Paul Kleneman group using both human adenovirus and chimpanzee adenovirus for their vaccine, which some of you might know that this is the same technology that has been deployed to develop the AstraZeneca Oxford COVID-19 vaccine. The strategy that this group used in developing the HCV vaccine focuses on parts of the virus that are less likely to mutate, which this is important for the vaccine to stay relevant against the virus. With the current SARS-CoV-2 variants that have been reported, the question is whether those approved COVID-19 vaccine design is capable of staying relevant to those variants. So this is something that more data need to be generated for us to see whether if those current vaccines are going to be still effective to those different variants as reported. So with the effort of identifying T-cell epitope, this has been characterized from individuals who had been managed to spontaneously clear HCV infection, which enabled this group to use this information to synthesize them into the MHC class 1 pentamers using the A0201 and A0101 restricted epitopes to track antigen-specific CD8 T cells in those vaccinated donors. So as shown in the staining data below, using the pentamers to stain at different time points of the trial subjects, they have shown that the population of antigen-specific CD8 T cells response sustained at about 1% at the end of the study, which is amazing to see that the anti-HCV responses induced were prolonged and broad with respect to the epitopes they were focusing on. In addition to this, the group in Oxford has done further immunophenotypic characterization of various biomarkers gating on antigen-specific CD8 T cells such as CD38, HLA-DR, PD1, CD45RA, CCR7, C127, and CD161. This list can go on with our pentamers for other biomarkers that you are interested to look at as well. This case study is something to definitely think about to monitor the immune responses of the COVID-19 vaccinated donors to look at the robustness and the effectiveness of the immune responses and characterize those T cells. Apart from monitoring the antigen-specific CD8 T cells, we should not forget about monitoring antigen-specific CD4 T cells, which is as important. Here is our Pro-T2 MHC class 2 tetramers that ProImmune offer, which comes in the labeled format in either RPE or APC ready to be used for experiment. The test quantities that we offer are either in 50 or 150 test quantities. We have about 180 specificities available in our catalog, of which 15 of them are viral hepatitis specific. And they are ready to be shipped out in about 1 to 2 days or 3 weeks. If you cannot find the tetramers that you are interested with, we are able to offer them as a custom tetramers for the human and mouse alleles available listed here. And they are ready to be shipped out in about 7 weeks. This product is 6 months guaranteed, and we can offer them in the biotinylated monomer format too. So here are some staining data shared by ELISpots group using our HCV tetramers on DRB1-1501 positive vaccinated donors. And then the top 2 panels are the staining data at 12 and 22 days cultured respectively. And the bottom data is using a HLA mismatch vaccinated donor as a negative control. So you can see very nice staining population of the antigen-specific CD4 T cells of about 22% and about 30% in these 2 respective staining data. And more staining data, so this one is shared by George Lauer from the Massachusetts General Hospital and has kindly provided this staining data using our HCV tetramer. And he has used 2 different specificities for the DR4 and DR1 restricted alleles. Yeah, and then for different HLA type donors for these 2 alleles. Yeah, so now I have good news to share with everyone today at this conference. So for this year, ProImmune has decided to reduce price for most of our products as we have improved our efficiency in production, which allows us to be more competitive. The prices are the same across all labeling options that we can offer, so you can select your preferred labels at the same price. Prices for catalog and custom products are the same. Not only that, we have removed the custom setup charge, so there will be no more additional cost for custom pentamers. That is a non-refundable component in the past. So moving to our services. So now you might be wondering what are some of the strategies out there that is available to identify epitopes and synthesize them into our pentamers. So of course, there are several different strategies that are used to identify epitope. And today I'll be sharing one of the service is called ProImmune REVEAL MHC Peptide Binding Assay, which is a high throughput screening assay to confirm the physical binding characteristics of peptides against MHC alleles, allowing you to map out potential T cell epitopes. We can offer both the class 1 or class 2 format for this assay, and here is the simplified workflow for our REVEAL MHC Peptide Binding Assay, where you will be providing us with the peptide sequences information, and we will run the physical binding assay in module 2 to look at the binding affinity of those peptides against those MHC alleles of your interest. For those hit peptides, we can synthesize them into either pentamers or tetramers for you to stain your cell samples using flow cytometry. Take note that peptides that have high binding affinity does not necessarily mean that they have good binding stability. Therefore, at the final step in module 4, we will incubate the MHC peptide complexes at 37 degrees Celsius for 24 hours to assess the stability of those peptide MHC complexes. So looking into HIV instead of viral hepatitis, here is a case study published by Imperial College London that have deployed ProImmune REVEAL binding assay and have successfully identified T cell epitopes for HIV-1. Since B3501 positive HIV-1 infected individuals are associated with rapid progressions, a robust CD8 T cells response is required to clear viral infection. We know that HIV gag is an important immune target, so if we could achieve a broad gag response, there will be a lower viral burden. In order for them to identify T cell epitope, they have used an in silico prediction tool where they have successfully screened out 44 peptides that are predicted to bind with B3501 allele. Since 44 peptides are relatively still quite a large number, they have decided to deploy our physical MHC peptide binding assay to narrow down further. So here we have synthesized those 44 peptides, and we screen them in our REVEAL binding assay. As you can see in this chart, 12 out of 44 peptides have shown to be good binders, but this does not mean that they are necessarily a stable binders. So in order for us to assess the stability, we use the quick check off rate assay, which is the module 4 for our REVEAL binding assay, which those 12 peptides incubated at 37 degrees Celsius for 24 hours and measures taken at 3 time points of 0, 2, and 24 hours. A quick score is calculated by having the REVEAL binding score multiplied by the half-life over 10,000. As you can see in the chart below on the left, peptide 8 has shown with the highest stability, thus they have decided to synthesize this into our Pro5 Pentamers and have validated this epitope in an ex vivo staining as shown in the right plot. So the one on the left plot is a negative control that they have done, and then the right plot is using the positive donor for them to do an ex vivo staining to validate this epitope for HIV-1. So in summary, for a successful epitope discovery and immune monitoring program, this requires detailed and efficient characterization of antigen-specific T cells epitopes for immune monitoring program. In order to achieve this, it is important to have access to a wide range of tools such as MHC peptide binding assay and MHC multimer technology to enable appropriate and rapid decision making. ProImmune is the world expert and leader in providing MHC multimers to our customers, which has been evidently shown in our publication track record citing ProImmune. We have the breadth of experience with more than 20 years of experience in providing the best-in-class MHC multimers, which can save you time, money, and reduce overall project risk. If you'd like to see more papers that have cited ProImmune, you may visit our website for a list of papers citing ProImmune in this web link here, and also highlighting our customer research in the second web link. Also, you can view those podcast videos that we have recorded during our past conferences by speakers that we have invited. So for today's conference, it will be recorded as well, and then if you want to view them on demand, we will be uploading them on our website, and then you can view them whenever you want to look at the videos again. So lastly, you can have a look at our product protocol to find out more about using our products in the last web link below. So I would like to take this opportunity to introduce our upcoming main event, Mastering Immunity Europe 2021 virtual conference that will be held on the 25th March, Thursday, 5 p.m. Singapore time. So if there's any other products or services that you are interested in to find out more, right? So which I did not manage to cover today, I'm happy to schedule another date and time for us to discuss in detail after this conference. So feel free to reach me out on my email or WhatsApp or our ProImmune mobile number in Asia. So here's my contact detail, and feel free to reach out. And if you'd like to find out more or you want to receive this slide deck, you can let me know, and I'm happy to send it over to you for this slide deck for you to view whenever you want. And before moving on to the last speaker of the day, I'm happy to take a couple of questions now. Yeah, thank you. So we have a questions asked by Antonio. Yeah, so Antonio asked what is the focus of the conference that we are organizing. I believe Antonio was mentioning about the Mastering Immunity Europe. So for this upcoming conference that will be holding in next month, 25th March. So this is actually a mixture of a conference whereby we have academia people, biopharma, and also the regulatory agency that is really joining us for this. So it's really to actually have people across the world to discuss about immunology in the space of looking at unwanted immune response or looking at unwanted immune response. So it's quite a similar kind of conference that we have held in the past before that happened last year September. So this time around we have invited a different list of speakers to actually share their work that they have done in the different space of their own project interest. Yeah. So I hope that answers your questions, Antonio. So it's quite similar to the conference that you have attended last year, but of course we have a new fresh list of people to speak and to share new data with us during the conference.